In the fields where our food is grown, a quiet revolution in genetics is changing how we protect crops. For 70 years, chemical pesticides have kept pests at bay, but at a cost: resistant insects, vanishing pollinators, residues that linger in soil and water. Agriculture needed a more precise tool. That precision is described in landmark research published in Advanced Genetics in 2026 by Dr. Ji-Xing Liu of Guangzhou University and Dr. Guy Smag of Guizhou University and the Vrije Universiteit Brussels. Their paper, RNAi-based pesticides, Genetic Innovations for Sustainable Crop Protection and One Health, maps the science that makes precision pest control possible. Their own Figure 1 captures the whole system at a glance: nanoparticle-delivered dsRNA, target gene discovery, spray and host-induced silencing, and protecting pollinators, all in service of biodiversity and One Health. For those who want the full pipeline: target identification and validation, dsRNA design and synthesis, delivery system development, application strategy, laboratory and field trials, regulatory review, and scale-up. 7 stages from a validated gene target to a farm-ready product. And the pathway in complete molecular detail: uptake of dsRNA, processing by dicer, risk assembly, target recognition, mRNA cleavage, and gene silencing, mapped alongside every delivery route and its downstream effect on the pest, the population, and the ecosystem. The silencing signal does not stop at a single cell. An enzyme called RNA-dependent RNA polymerase amplifies the initial trigger into secondary siRNAs, which spread cell to cell and travel systemically through the pest, reinforcing gene silencing across the whole tissue. Taken as a whole, this is one integrated system: precise gene silencing, multiple delivery innovations, multi-omics accelerated discovery, and a direct line to One Health: vector control, pollinator protection, environmental safety, and human health, together. At its core is RNA interference. A double-stranded RNA molecule, matched exactly to a pest's own gene, is cut by an enzyme called dicer into small interfering RNAs. Loaded into a protein complex called RISC, these fragments seek out and destroy one, and only one, genetic target. Liu and Smag describe 2 ways to deliver it. Host-induced gene silencing engineers the crop itself to produce the RNA. Spray-induced gene silencing applies it topically, no transgenic plant required, and a lighter regulatory footprint. In the field, spray-induced gene silencing looks remarkably ordinary: a targeted application, absorbed by the crop or the pest directly, carrying its genetic instructions to exactly the insect it is meant to control. Because naked RNA breaks down quickly in the field, their research highlights nanocarriers, chitosan, lipid particles, star polycations, clay nanosheets, each extending the molecule's stability and reach. Sensitivity varies by insect order. Beetles respond strongly, moths far less so, which is exactly why specificity matters. The same precision that eliminates a pest can protect a pollinator. Commercial RNAi miticides like Noroa already shield honeybee colonies from Varroa destructor, extending this science into One Health. Their paper is candid about the risks too. Resistance can still emerge. Rotating gene targets and stacking multiple independent targets are the safeguards that keep the technology durable. This isn't theoretical. SmartStack's Pro Corn and Greenlight Biosciences Calantha are already protecting fields today, proof that RNA interference has moved from laboratory to farm. This body of research is exactly what led to the creation of RNAi Precision Crop Designer, an intelligent operating system for designing, evaluating, optimizing, and deploying RNAi-based crop protection strategies. It turns Liu and Smag's science into a working design tool, scoring candidate sequences, screening for pollinator safety, and simulating knockdown, persistence, and resistance, all in one place. Built on published science. Designed for precision agriculture. RNAi Precision Crop Designer, where genetic insight becomes a deployable strategy.